We're going to begin an exploration into the Duchenne research panels, and this is our series called In the Pipeline. This first In the Pipeline discussion is reducing inflammation. So we're going to hear from companies that are focused on developing therapies aimed at reducing inflammation. And Eric, perhaps you can explain a bit about, Eric Camino, about this mechanism of reducing inflammation and what these drugs are meant to do.
Yeah, so I think we're all very aware of the fact that inflammation is a big challenge within this disease. When we think about the root cause of this disease being a mutation in the DMD gene and that causing a lack of production of dystrophin, that ultimately contributes to these weaker muscle membranes. And so what happens, we have contents from the cell that escape and are seen as damage signals. And so your immune system is going to respond to that. It's going to create this and promote a pro-inflammatory state. And within those who have Duchenne, we kind of end up in this state of chronic inflammation. And this has negative impacts on a lot of the parts of the body and can even contribute to some of the muscle damage we see. So because of that, there are different strategies that we have to try to tackle this problem of inflammation. So we can try to, modulate the immune response or target pathways that are involved in that inflammatory response. So we're going to hear about a couple of different investigational products that are looking to reduce inflammation, as well as one of the approved products that we have in Duchenne. And so today, we are going to be joined by Dr. Eric Hoffman, who is the VP of Research at ReveraGen. We're going to be joined as well by Dr. Joanne Donovan. She is the Chief Medical Officer at Catabasis Pharmaceuticals. We also have Dr. Brian Pfister, who's the Executive Director and the U.S. Medical Head of Neurology at PTC Therapeutics. And we will have Dr. Gil Price, who is the Chief Medical Officer at Antisense Therapeutics. And so, as a reminder to everyone watching, if you have any questions while the presenters are going through their 6-minute presentations, please put them into the comment box below and indicate which sponsor you're directing that question towards, and we will address those when we get to the Q&A session. So we're going to go ahead and begin with Dr. Hoffman, who's going to tell us a little bit about vamorolone. Dr. Hoffman?
Hi, thanks. Pleasure to be here and speak with you. So I'm here representing Robergin, and the drug we're developing is called vamorolone. It used to be called BBP-15, and it's meant to replace corticosteroids. So in doing that, it also targets the glucocorticoid receptor, which is the main target of deflazacort and prednisone. But it also targets the mineralocorticoid receptor, which is similar to another drug already used in Duchenne for heart failure called eplerenone. There's also a membrane stabilizer. So it has multiple mechanisms of action, some of which are shared with corticosteroids and some which are quite different. And in the left there, it shows just the X-ray crystal structure of it interacting with the receptor. So we know exactly the differences, for example, between prednisone, vamorolone, and deflazacort. Next slide, please. So DMD is the initial indication. Corticosteroids are among the most prescribed drugs worldwide. It's one of the top 3 drugs, I believe, prescribed for everything from asthma, inhalers, to all sorts of things. Duchenne, so vamorolone might be used in many disorders, but Duchenne is the first disorder we've gone forward with, and we have connection with many foundations, including Parent Project, in developing this. It's orphan, we have expedited development. It's driven by a international team of DMD academic leaders. We used an established clinical trial network called Synergy. How do we make that bigger? And the goal with all of these is to then take what's good with corticosteroids, which is the benefits that you see on the sort of left side of the scale, and keep those or improve them, while there's a number of safety concerns. Can you go back to the last slide? Number of safety concerns on the right, including bone fragility, mood changes, weight gain, and we'd like to reduce those. Next slide. So here's some, we had 48 kids finish 2.5 years of treatment with vamorolone, which was the first Duchenne trials. And up in the upper right, you see, for example, stunting of growth. So with vamorolone, we see normal growth patterns with Duchenne children, even after 2.5 years or more of treatment, where with deflazacort or prednisone, there's stunting of growth. So it keeps the kids from growing normally. So that's one safety concern that we seem to have avoided. In the lower left is comparison of some physician-reported safety concerns, such as cushingoid, which is the bloated appearance, hirsutism, which is extra growth of hair, behavior changes. And you can see with all these, the physicians report a higher frequency of these with deflazacort and prednisone in 2 published studies compared to vamorolone. So vamorolone seems to be a quite safe drug. And again, we've had 48 boys now all have finished 2.5 years, and many are 3.5 years and more now. And we've had 0 serious adverse events due to vamorolone. Next slide. But more important is potential benefit. So do we see efficacy, and is that similar to, or better or worse than corticosteroids? So one way to look at this is time to stand. So time to stand from the floor is one of the first skills or abilities that's lost with age in Duchenne. And on the upper right, we see the line in the middle is 5 seconds. So the kids that are faster than 5 seconds generally are stable or actually will improve with age, and that's known through synergy studies, Craig McDonald, and other large studies. Whereas if you're slower, if the boy is slower than 5 seconds, they're likely to decline in the future. And so when these boys entered the vamorolone trials at 4 to 7 years of age, many of them were slower than 5 seconds, and some were faster. Then after 1.5 years of treatment, you see almost all the kids became faster than 5 years. So this did much more than stabilize the disease. All the kids seemed to, or most of the kids improved even after 1.5 years of treatment. So it seems to be roughly the same benefit. We were doing a direct head-to-head comparison in the current trial, the prednisone. And we've had high satisfaction of physicians and families, and most of those 48 kids have gone on to expanded use and expanded access and compassionate use. Next slide, please. So in terms of regulatory, we're currently fully enrolled in a phase 3 or phase 2B, it's called, but it's our registration study. Readout should be in February 2021 after 6 months of treatment. The target age to date has been 4 to 7 year old steroid naive Duchenne boys, but we have started funding for and plan to kick off a broad age range from 2 to 4 years old and 7 to 18 years old. The older kids can also be previously treated with corticosteroids and transition to vamorolone. Our primary endpoint is improvement in time to stand from the floor, and we expect to have FDA submission late next year and EMA about 6 months after that. As I mentioned, this high family physician satisfaction with vamorolone. 94% of kids, the 68 kids that have come off trials to date, have all asked to stay on vamorolone, their families and physicians, rather than transition to standard of care, which is deflazacort or prednisone. Next slide. And we'd like to thank all the foundations that made this possible. We've partnered with about 15 different foundations internationally, including Parent Project, who's been instrumental in supporting these studies. Thank you. Back to you, Ryan.
Wonderful. Thank you so much, Dr. Hoffman. I'm going to ask you one follow-up question before we move on to the next speaker. In regards to broadening that age group, which we think is great, will patients just need to wash out of, if they're on steroids currently, of their current steroid regimen to join that type of study?
That's a great question. And I can tell you that's elicited enormous amounts of debate within the study team, including bringing in endocrinology consultants from around the world. And, there is no consensus on that. The whole weaning of steroids has very little scientific medical evidence behind it. It's more common practice than it is established evidence-based medicine. So as the trial's currently designed, we're going to go day to day. One day, the last day of prednisone or deflazacort to the next day of vamorolone. In part because vamorolone should replace some aspects of the concerns regarding stopping glucocorticoids quickly. Mm-hmm. So I think, for example, adrenal crisis, perhaps vamorolone will protect against that.
Mm-hmm. Wonderful. Thank you so much, and I'm sure others will have questions during the open Q&A. We're going to move on to our next speaker, Dr. Joanne Donovan, who's going to talk about etasolinexant. Dr. Donovan?
Hello, everybody. My name is Joanne Donovan. I'm the Chief Medical Officer at Catabasis. Thank you to PPMD for the opportunity to share information today about etasolinexant and our approach to developing a treatment for Duchenne. Next slide. Catabasis is a publicly traded company, and during this presentation, I'll be talking about things that may happen in the future. Next slide. Etasolinexant is an oral drug in development for potentially everybody affected by Duchenne, regardless of mutation type. It's an NF-kappa B inhibitor, and it's not a steroid. We have data supporting etasolinexant for treatment alone, and we also believe that etasolinexant could potentially be used with other treatments, such as exon skipping and gene therapies. We've seen that etasolinexant preserved muscle function and substantially slowed disease progression while being very well tolerated in the phase 2 MOVE-DMD trial. And we're currently studying etasolinexant in our ongoing phase 3 Polaris-DMD trial and the Galaxy-DMD open label extension trial. Etasolinexant is an investigational agent, not currently approved in any territory. Next slide. Etasolinexant inhibits NF-kappa B, and we designed it that way because of the potential for broad therapeutic benefit in Duchenne. When boys with Duchenne are born, they lack dystrophin. However, it often takes a couple of years before symptoms are noticed. What happens in this time period? It's the absence of dystrophin and mechanical stress in the muscles that leads to chronic NF-kappa B activation, leading to inflammation and fibrosis and creating a cycle of damage. So our intention by inhibiting NF-kappa B with etasolinexant is the goal of improving skeletal muscle function, preserving cardiac muscle function, and also improving bone health with a decreased risk of fractures. Next slide, our fully enrolled phase 3 trial, Polaris-DMD, is a 12-month trial with etasolinexant for boys with Duchenne aged 4 to 7 up to their 8th birthday. Now, Polaris-DMD is a placebo-controlled study as needed for the FDA, with 2 boys receiving etasolinexant from the beginning for each boy that receives placebo. After 12 months, all the boys have the option to receive etasolinexant in the open label extension study, Galaxy-DMD, along with their eligible siblings. We enrolled boys that have not been on steroids for at least 6 months, as there is the potential for partial overlap in mechanism between etasolinexant and steroids. We expect to have results from this phase 3 trial in the 4th quarter of this year. Pending positive results, we then plan to file these results with the FDA in 2021. I also want to mention Galaxy-DMD, the open label extension study, which means that everyone participating in Polaris-DMD at that point would receive etasolinexant. We had heard from families that had sons who participated in the phase 2 study, MOVE-DMD, that they have other sons affected by Duchenne and wanted them to receive etasolinexant, therefore enrolling eligible siblings into Galaxy-DMD. Once boys complete Polaris and continue on into Galaxy, the eligible siblings will have the opportunity to enroll then. The primary focus of Galaxy-DMD is to continue to evaluate long-term safety in boys. Due to the excellent safety profile of etasolinexant that we've seen to date, site visits in this study are every 6 months. On the next slide, we'll briefly review past results we've seen in the phase 2 MOVE-DMD trial, which enrolled 31 boys aged 4 to 7 to their 8th birthday, not on steroids. The phase 2 MOVE-DMD trial had a built-in internal control where we assessed boys off treatment and then on etasolinexant. Before they started on etasolinexant, they were declining, and on etasolinexant, the largely flat green horizontal line shows that boys had preserved muscle function and stabilized their disease progression through 72 weeks on etasolinexant compared to the off-treatment control period for the North Star Ambulatory Assessment. We also saw similar stabilization with etasolinexin treatment for the 3 times function tests, the 10-meter walk-run, 4-stair climb, and time to stand. On the next slide, in more than 150 years of cumulative patient exposure, we've seen that etasolinexin was well tolerated. The majority of adverse events were mild in nature. The most common treatment-related adverse event was diarrhea, generally mild and transient. We saw no SAEs, serious adverse events, on treatment. We are also pleased to see that boys on etasolinexin grew like their unaffected peers, gaining height over 72 weeks and a small amount of age-appropriate weight. As etasolinexin is not a steroid, we haven't seen and don't expect to see steroid-associated side effects. On the next slide, to close, I'd also like to mention that we announced a partnership with Duchenne UK to study etasolinexin in boys and young men that are non-ambulatory. We'll share more information about this potential phase 2 study in the coming months. I'd like to thank you for the opportunity to share this information with you. And for those of you who are interested in seeing updates along the way, we share frequent updates on social media, and you can follow us on Facebook, Instagram, or Twitter. Thank you.
Great, thank you, Dr. Donovan. I do have one follow-up question before we move on to that next speaker. You had mentioned etasolinexin in combination with exon skipping. You know, we now have 2 approved exon skipping drugs, one of which I assume may have been in patients when they went to actually recruit for your trial. Do you have patients on Exondys involved in your trial, and are you looking at an arm of those patients with that combination to see if there's a specific effect?
Yeah, that's important because we think the future is combination therapy, that lots of boys would be on that. So actually, in all 3 of our clinical trials, we have boys that are on combination with exon skipping therapy. When Exondys was approved, we amended the protocol to allow boys to start, and boys in the phase 3 trial were randomized based on whether or not they were on Exondys. With the advent of additional therapies in the extension study, we're also allowing boys to start on golodirsen or other drugs as they may become available. That's
great. Thank you so much. We so appreciate it. Okay, we're going to move on to the next speaker on our panel. This is Dr. Brian Pfister with PTC, who's going to talk about Emflaza, which is the only approved drug of those on the panel. Brian?
Great, thank you. I appreciate it. As always, it's a pleasure to be here. I love working with PPMD, and we've been working for a while, so definitely appreciate it. So when you look at what we're doing, we're trying to understand, the history that PTC has within Duchenne is incredibly long, and it gives us an advantage with regard to understanding what's happening with these boys. On the next slide, as like other companies, we are publicly traded, so here's our forward-looking statement. So on the next slide, we'll kind of get into the meat of what we're talking about. So when we look at this, and as had been mentioned earlier, so if we look at what's happening with regard to the role of inflammation in Duchenne, as was mentioned, we have this dystrophin deficient muscle, which has this leaky membrane. So it's leaking this material out that the immune system sees as damage, sees as an issue, and this kicks off the inflammatory cascade. So once the immune system recognizes this signals cytokines to produce downstream effects in this inflammation that was talked about earlier during the intro, signals T cells and B cells both in a way that creates this constant inflammatory state. So we're able to identify targets within this space. It's a very target-rich environment. We're able to identify targets within this space as therapeutic opportunities. So what we've seen within steroids, they work very well because they hit multiple different targets. So on the next slide. When we look at this, when we look at the T cell in particular, so this is important because as we mentioned before, when the leaky membrane is presenting this muscle tissue or presenting this muscle as an antigen, it does trigger an immune response. So when we look at prednisone, we look at deflazacort, we look at these kids who have this immune response, we do see a differential effect between these 2 treatments. When we look at this, we see that the deflazacort kids tend to have a less reactive immune system to dystrophin as an antigen. What this means is it may suggest in some way, shape, or form that we're actually controlling inflammation differently. So as you know, Emflaza, like Ryan said, is the only approved product that we're talking about today, approved for ages 2 and older. It's the only approved corticosteroid on the market, I should say. So when we look at this and we understand interrupting things like interferon gamma, which is this T cell response, has a downstream effect. So on the next slide, we can see exactly what that effect does. So by sitting on the interferon gamma receptor and changing the way that the interferon gamma is signaling, we hit multiple pathways in the inflammasome. So newer therapies, newer treatments may only target one particular side of the pathway, leaving the other side of the inflammatory pathway open to continue to create inflammation. So this suggests that there may be issues with regard to the long-term effectiveness of some of the newer treatments. So if we look at the next slide, when we have a long history of treating with steroids. And so we're able to see what the side effects are. We're able to understand those. We're able to understand too, even though if we're modulating the immune system differently, we're not seeing this increased risk of infections. We're not seeing an increased risk to the patients in general. So the benefits clearly then outweigh some of the side effects that we're seeing. So the common ones that we do see are obviously weight gain, behavioral changes, and we do see differences between deflazacort and prednisone in these areas. So this tells us, again, there's always going to be side effects. If you're not seeing side effects, there's always the question as to whether or not your drug is working. And what's important to look at here too is when you're comparing steroids, you're comparing drugs, you have to compare them within your study, because there's age differences. So for example, deflazacort has been studied in much older children than some of the newer products. So if you look at the side effect profile, you have to keep that in mind, that patient populations are different, inclusion criteria are different. So on the next slide, when we get into what the benefit is and looking at the long-term benefit, I think this is really where it's important, because endpoints are really, again, what we're studying during the course of a trial, endpoints have to be looked at in long periods of time. So if you think about this in a decade-long study, we can actually see this benefit and we can see this impact of treatment to these kids. So for example, we talk a lot about loss of ambulation. And if you look at this, if you were looked at where the 50% mark is for prednisone at the age which half of the kids in this particular trial lost ambulation, if you correlate that then to where deflazacort is, you have 50% lose ambulation, about 3.5 at that, or at 13.5 at that same age, at that same point in time, you have about 85% of the deflazacort kids still walking. So this is a very meaningful endpoint. And this is very important to look at, because if you look at the age ranges within this graph, you see that kids don't begin to decline until after 8 or 9 years old. So studying it very early, you may not see the same decline in a long period of time. And what's even more important, I think, when you look at this, is to look at the scoliosis onset. So things that are tied into muscle function in the long-term benefit, like scoliosis, I think is something that deflazacort has this advantage of, a very meaningful endpoint to a lot of families. So on the next slide. When you look at this too, and I want to highlight pulmonary function, because again, this is an incredibly meaningful endpoint, because when kids can't breathe, they can't live. So if we look at this and we understand that deflazacort has a difference from prednisone with regard to functionality of lung tissue, this is really, really important. And I think that what you won't see in the newer studies and the newer drugs being developed is you're not going to see this benefit, because it does take a while to see this. It takes, again, over a decade and a lot of times to see this. So I think these are important points that we can't overlook in drug development. And I think it's something that we just need to keep in mind in general. Pardon me, getting choked up about it. So on the next slide, if you look, again, kind of in closing, we have learned a lot about steroids. We've learned a lot about deflazacort. And what we've seen is deflazacort consistently is better than prednisone. So when deflazacort's not being used in new clinical trials as an active reference or comparator, I think we're missing the boat. I think as a community, we need to come up with better treatments that are moving the bar forward, not just looking at it compared to prednisone. So thank you for your time.
Great. Thank you, Brian. Just one quick follow-up. In terms of now tracking patients who are on the approved therapies, are you all, do you have a registry that you're looking long-term with patients on Emflaza? We know that insurers require paperwork, et cetera, but is there a national sort of registry across the patient population on Emflaza?
So for Influenza, we don't have a registry that we're working on. We do work very closely in the medical community. So, as I've mentioned before, working with you guys so closely, it feels like we know the patients by name. Yeah. So in working in the field, we have that same relationship with a lot of clinicians. So we don't have a registry, but we're well aware of the activities in the field.
Wonderful. Yeah, and that includes, we working with us with our registry, we're able to track patients on your therapy. So I think there's a lot of opportunity for collaboration. Okay, we're going to move on to the next speaker. Thank you, Brian, so much. Our last speaker of the panel is Dr. Gil Price, Chief Medical Officer with Antisense Therapeutics. Dr. Price.
Thank you very much for having me. And thank you, Pat and PPMD. We're grateful that you allow us this time. It's also good to be on any panel with my old friend, Eric Hoffman. Let me tell you just a little bit about Antisense. Antisense is a small but publicly traded company in Australia. Our lead candidate, and by the way, we're focused on rare disease and in particular Duchenne. Our lead candidate in this area is ATL-1102. It is a drug that is designed to inhibit CD49. CD49, I will tell you a little bit about in a moment. But first, let me tell you about the study that we've recently completed, or just a wee bit about the study completed. It is a study in non-ambulant boys, 12 to 18 years of age, with the median age of 14. Before we get into other details of the results of that study, again, let me just share with you a little bit about the drug. ATL-1102 is a 2-MOGAPmer antisense oligonucleotide. It's designed, again, to inhibit CD49 expression on lymphocytes. Interestingly, DMD patients have greater numbers of circulating T cells with high levels of CD49 expression. And that sort of gets to the mechanism of action that we believe that we can allow our patients to benefit from. Also, interestingly, we don't believe that corticosteroids appear to have any effect on CD49 T cell numbers. So again, we think that that's something that gives us a bit of an advantage. Next slide. So just getting to the results of our phase 2 study, we were able to meet our primary endpoints and had strong effect on our secondary endpoints, activity on our disease progression markers. We had improvement and stabilization across different measures of motor function and strength via performance of upper limb tests, and pinch strength and distal mobility. Our activity on the targeted CD49 cells was consistent with what we believe is the drug's mechanism of action. And now we've just recently had new MRI data that suggests stabilization of the percent of fat in muscle, which we believe is very important. We're happy to say that we have support from key international opinion leaders, which takes me to the next slide. Are just the key takeaways. Thank you. The key takeaways that I would leave you with. As the last presenter, I'm trying to get off as quickly as possible to allow you to move on. So the key takeaways would be this. Our phase 2 study has shown that ATL-1102 is a treatment that resulted in consistent improvements or stabilization across a number of different measures. We have support from the DMD community. And right now, not surprisingly, we're looking into our strategies for moving forward in a global way. What can we do from a regulatory study design perspective? And those discussions are going on internally, and they take a lot of our time and energy. But we know that it's worthwhile. We want to get ourselves to the best place possible to help our boys live longer and better lives. So, Ryan, I'll leave it with that and send it back to you.
Wonderful. A quick follow-up on this. I'm sure that you've been asked this before and everyone's asking it. Timing for coming to the U.S. Is there a rough estimate or do you have a sense of when you will be planning studies?
Right. So again, we're a small company in Australia. represent, along with Aaron Cox, and two of our board members. The four of us represent our presence in the U.S. currently. But we believe that we may be small, but we're strong and well intended. So we're encouraging our senior management in Australia to think about coming to the U.S. More quickly than they may have ever thought. So I can't give you an exact date for that, but we are eager. We believe that we have good support here, and we intend to be here sooner rather than later.
Wonderful. We like eager, and we appreciate that you're in the space, so we will be keeping in touch with you. Okay, thank you so much, Dr. Price. We are now going to move on to the Q&A panel. Lots of questions have come in since you all started speaking, and Eric's going to lead that Q&A. Eric?
Yes, and so please remember, if you do have questions, submit them to that box below, and we will try to get to them. Those that we can't, we will pass on to the sponsors. But to begin, so we've got a lot of questions running on similar themes, and so I'm going to start with one that has been coming through a lot, which with the investigational products in the relationship to steroids. Can you just clarify, do you anticipate this being taken in place of steroids, which we know are standard of care, or in combination with steroids? And so, Dr. Hoffman, I'll start with you.
Yeah, it would be one or the other. It would not be both at the same time.
Dr. Donovan?
We see this as a foundational therapy that other drugs like exon skipping can be built on, but the foundation.
Okay. And Dr. Price?
So similar to that, we would see this as a foundation that other drugs can be added to. And as I sort of tried to highlight, we don't believe that steroids have any effect on CD49. So while we may have that profound effect on CD49, steroids may be accretive to that on the other side.
Okay, great. We also have a lot of questions coming in about how these products may interact or be used alongside gene therapy. So we do know that steroids are still currently used early on in the process, and those boys tend to still be on them. So can go around to all of our presenters, and actually, Dr. Pfister, I'll start with you on how deflazacort may be used with gene therapy.
No, I think this is a great question, because when you look at it and you go back to, again, Eric, what you had mentioned about the dystrophin being present within the leaky muscle tissue. So when I replace dystrophin or mini dystrophin, I actually still have to control for that immune response. So this is going to be absolutely critical to this. And when you look at the life cycle of T cells, they regenerate every day. So when you look at even the idea of daily dosing of steroids to protect that newly produced dystrophin that you have via the immune or via the gene therapy is critical to the long-term success and functionality. Also, switching that M1, M2 to be regenerative is going to be critical to that piece. So I see it as still a cornerstone.
Great, thank you. Dr. Price?
So it's a little bit early in the discussion for us to be thinking about that. But again, I can see a path forward. So I'll just leave it at that for the moment, considering where we are.
Okay, great. Dr. Donovan?
Yes, so in gene therapy, there's two different ways that steroids are used. And they're used initially to suppress the immune response to the virus itself. That's high dose. And any gene therapy, that is the case, that there are several months where the boys or whoever is on high dose steroids. Then long term, do you need steroids is the question, because with other gene therapies, you're able to taper off at that point. And because the goal of gene therapy is to convert what looks like Duchenne to something more like Becker, there still is believed that there will be inflammation there. So ideally, you would have something long term in the case of Duchenne to maintain and to continue to suppress inflammation. So we think that there's kind of two different questions. What do you use short term, and what would you use long term? And we think that for long term, that has great potential for edaselenexant.
Great. And Dr. Hoffman, on that note, do you think, in addition to maybe the long term, that there's a possibility for vamorolone to be used in that early kind of immunosuppressant phase? Did have a question about that as well.
It's possible, but yeah, I think that early immune suppression stage in gene therapy is extremely important, and where kids run into trouble too, side effects that hospitalize them. Not just in Duchenne, but in many disorders. So everybody's trying to tweak that immune reaction with multiple drugs, but even besides steroids. But I agree with Joanne that our place is certainly in the would be used. Vamorolone would be used in combination with gene therapy long term. The acute is much trickier, as Joanne said.
Thank you. And actually, we have another set of questions coming on, and Dr. Donovan, I think you kind of hit on this already, just in general about possibility for these drugs to be tested or for the labels to be expanded to include patients that have Becker muscular dystrophy, or for those female carriers that may manifest symptoms early on with some more severe progression. So if you could all maybe share what your thoughts and plans are for those other diagnoses. And Dr. Donovan, I'll start with you.
Yes, I think that Becker is a disease that has not traditionally been treated with steroids, although some of the patients who have more severe symptoms are. But and that's because of the side effect profile of steroids. So if you could suppress the inflammation, the NF-kappa B driven inflammation without the negative effects, that would be a very good thing. So that is something that we are thinking about. And there's actually even other forms of muscular dystrophy in which inflammation is key.
Great. Dr. Hoffman?
Yes, the National Institutes of Health is just funding a clinical trial of vamorolone in Becker muscular dystrophy out to Paula Clemens at University of Pittsburgh. Mm-hmm. Plan to start that soon. Another advantage of vamorolone in Becker is we've shown that it increases dystrophin levels in Becker models. So, and that this has to do with microRNAs and so we hope that vamorolone will show even better efficacy in Becker than it does in Duchenne.
Great, thank you. And Dr. Price, I know you said this is still very early days, but maybe you can share your thoughts.
Yeah. So keep in mind that our phase 2 study was in non-ambulant boys, which is an area that there are not too many players in. And so that, again, was a reason for encouragement after we took a look at the very positive data. What we've talked about now is not necessarily Becker's, but what we've talked about is expanding into ambulant boys as well. Following along with that, you could certainly see how we may end up down the road in that Becker's model as well. So I wouldn't put that aside, but I think our next step will be into the ambulant population.
Great, thank you. And Dr. Pfister, obviously, as Dr. Donovan indicated, we don't want to necessarily put Becker patients on long-term use of steroids. We did have a question come in though about use of Emflaza. If you have any data looking at kind of the different dosing regimens, so whether daily dosing or weekend dosing, if you guys have been able to collect any data on that.
Yes, there's actually, there are data that do exist on that. So up in Canada, Doug Bigger had done some research on that. And so there is a study that shows that. And what you see is anytime you have a steroid, again, getting back to the T and B cell story about how this is working, anytime you're dosing a steroid in an alternate dosing fashion, you're going to not have the same effectiveness with regard to the T cell modulation. And so what we do see is in the Synergy study, you saw kids who had alternate dose prednisone. So kind of getting to that, you did see a difference in their effectiveness as compared to daily. So in what we saw within the, what Doug had published, within that space is that he looked at alternate dosing in a different form with Emflaza or deflazacort actually in that case. And what they did see is that there was some benefit, although it's decreased. And that is consistent with what you see. And you have to look at this over the long term again, because things in the short term sometimes do look comparable. Long term, you see vast differences. So,
great, thank you. And I know, obviously, or we all know that steroids are used throughout. the course of the disease. And Dr. Price and Dr. Hoffman, you both indicated your current plans for non-ambulatory. Dr. Donovan, could you maybe touch on your plans for non-ambulatory? I know there was an announcement shared, I believe it was earlier this year or maybe late last year, about plans for non-ambulatory.
Yes, Duchenne UK graciously granted us funds to initiate a study in non-ambulatory, and so we are working on plans for that right now in the non-ambulatory population. Great.
No, we love to see the expansion to make these products available to all members of our community. We had a question come in asking about collection of CK levels, and I think we can maybe expand this a little broader. Of in addition to the functional outcome measures that you are all collecting, what potential serum biomarkers are you looking at, and are there plans with any of those type of collections or anything from the placebo groups that you are running to share that data with the community? So maybe first I'll direct this one to Dr. Hoffman.
I think biomarkers are very important. Everything in biomarkers is what they call context of use. How are you using the biomarkers, and then making sure the biomarker is fit for purpose. And we've used lots of biomarkers throughout the vamorolone program in different contexts of use. Specifically with regards to CK, we see a dose-dependent decrease in CK, highly significant over 2 weeks. So, and that, even when you stop the drug, persists for another 2 weeks. So we are seeing what we think is the membrane stabilization and loss of inflammation. The CK shows that as a biomarker. Although if you look long-term, one of the problems with CK is it's extremely volatile. So, you start exercising or not exercising, it starts changing your CK. So CK is not a particularly reliable biomarker. We were actually surprised we saw such reductions. But you've seen it also impressive changes in gene therapy and other disorders. So we're using other biomarkers like inflammation biomarkers, membrane stability biomarkers, and we're using those both acutely, long-term. Our context of use is often to try to find the right dose because they become what's called objective outcome measures. You can watch them change very quickly in the blood to know that the drug's hitting its target, and that's how we use them mostly. And we make all that data public.
Great. Again, we love to see the sharing of data. Dr. Donovan?
Yes, we have looked at biomarkers like CK, and we've seen CK and other enzymes as well as biomarkers reduced statistically significantly. We've also looked at MRI, which is a really great biomarker in terms of looking at muscle, the structure of muscle. And we're able to see changes there, working with the imaging DMD group, again, seeing statistically significant improvements there. And ultimately, yes, I fully support data sharing as we move forward in the placebo group. There's a lot of great data that has come out of those data collaborations, de-risk, CTAP, and a lot of important learnings in terms of understanding the natural history when you look at data across placebo arms of many studies.
Great, thank you. Dr. Price?
Yeah, I agree with Eric and Joanne about data sharing and the value that can come from that. I also agree with Joanne. There's a lot of value in MRI data. For example, as I mentioned, we were able to see how fat was spared in muscle with our drug. That's an important piece. We're also taking a look at various biomarkers. I do agree with Eric. CK is can be a little squirrely. So we're trying to take a look at what other interleukins and cytokines that may be able to give us a little clearer picture. But we'll be doing that and making that a part of our 2B follow-up study.
Mm-hmm. Thank you. And Dr. Pfister, can you maybe share some of what PTC has done to date?
Sure, definitely. So one of the things about the Deflazacort that is nice is that we do have an understanding of the cytokine signature, right? And so the important thing within Duchenne is what it is at 5 is not what it is at 15, right? So there's this fluctuation. So it's like a fingerprint. It may be similar, but there are variations based upon what the journey of the patient is. So we've been able to isolate different cytokines, understanding the role of interferon gamma, for example, IL-10, IL-6, TGF beta. So as we get into this, understanding exactly what this looks like, we can then predict where we should be. And from this, then we can begin to spring forward and think about what other molecules could be developed based upon why we've seen such good efficacy and long-term effectiveness with Deflazacort.
Great, thank you for sharing that. And then one quick last question, I think, before we wrap this session up, given that we just had that really great discussion earlier in the day about COVID-19, I wonder if you could all maybe share something that you've adapted in your trials or, with PTC, how maybe that's impacted your decision-making process as we're kind of in this new landscape. And so actually, Dr. Pfister, I'll start with you.
Yeah, so it's definitely something we're looking at because I think everybody's been impacted with the timelines, right? So we'd love to be further ahead than we are in certain things, but what it has really signaled to us is as we develop new medications, they have to be tolerable and effective, and they can't be sensitive to change, right? So as we can't get out and see physicians, I think it becomes very important. The other thing too is it really puts focus on with telemedicine, the ability to use upper limbs, right? So when you think about this, so you build in maybe some of these as endpoints in clinical trials, looking at is their upper limb function maintained, because as these boys grow into men, if they're 18, they can go to the doctor on their own with telemedicine. So there's this huge idea, let's think outside the box. How do we connect people to the information they need, and how do we maintain the ability to be independent? So we've looked at that, and we're really focused on what that means to the future of development.
Thank you. Dr. Price, we know that you're, early on in some of the planning stages. Has this impacted some of the trial design that you and the group at Antisense Therapeutics are considering?
Yeah, well, it certainly has. I mean, I don't know if you are aware or saw in the European Journal of Heart, they just did an observational look of just over 1,200 subjects who were COVID positive, and 55% of those people that they looked at had an impact on their heart, had cardiac changes, 55%. So it started to make us think about, gee, if that's true for heart, can it be true? Does it have implications for Duchenne? And how we take a look at our endpoints for potentially boys who have been COVID positive. Along too with what Brian is talking about, I mean, we're thinking about all those kinds of things. How can we employ telemedicine, wearables, Bluetooth thermometers, et cetera, to collect the data that we need against this backdrop of the pandemic? So yeah, it's a part of our conversation.
Great. No, those thoughts about, what can we do to make things a bit more remote and open it up, and potentially for parts of our community that don't have access to clinical trials, those are great ideas to hear. Dr. Hoffman, how has ReveraGen been adapting?
Well, certainly the word that everybody seems to use is pivot. You feel like you're pivoting so much, you're dizzy. But yeah, we had to rapidly move to remote assessments, and because patients couldn't travel or weren't permitted, research subjects weren't permitted to go to their clinical sites. So things are back in most places, but then there's dangers of them being shut again. It's also made us, it sort of changed the regulatory landscape a bit and brought a bit more leniency into it. So, what we've seen is really, do we need so many clinic visits during a trial? Because, there's not only is it burdensome to the families and also expensive, but at some point it's a risk, with COVID-19. And so that changes that set point of monitoring safety, where maybe less monitoring is more safe, and particularly remotely. And that's an interesting change. And then finally, I'd also echo the remote assessments. So Roxana Ben-Dixon at Pittsburgh is running a sub-study on our pivotal trial ongoing, where we have actigraph monitors 24/7 over a year on 2 to 4-year-olds in our trial. So we have all their sleep health, we have their activity, and then we can correlate those with what happens in the clinic and those assessments. And the goal is maybe these actigraphy devices can start replacing clinic visits.
That's a really exciting addition. I'll be looking forward to seeing that data whenever you're able to share it. And Dr. Donovan, again, thank you for participating in our earlier conversation about it. We really appreciated your insight. Is there anything you'd like to add as well?
I think that, certainly it's changed a lot of the ways that we do things and moved things ahead to use telemedicine, remote assessments. I think the very impressive thing that we have seen is we're all in this together. We have just seen the families, the sites personnel. Sometimes they're deployed to other areas. They're still managing to get stuff done. So it's really an incredible effort by the whole ecosystem to move things ahead and to make sure things continue to work. So thank you for all everybody out there who has been helping on this.
Well, thank you. And thank you to all of our panel members. We really appreciate your participation today and answering those questions from the community. For those who didn't get their questions answered, we are going to be sharing those with the sponsors. And as a reminder, you can go and visit all of these companies' presentations, a little longer form, in the conference hub. So you can learn more about any one of these products. And again, feel free to submit your questions there.